Hello, dear students. I welcome all of you on this platform. In this lecture, we'll discuss about your first paper, that is microbial techniques. In this, we have to discuss about three different units. The syllabus includes sterilization and disinfection. This is nothing but the basic introductory part of your syllabus, in which we have to discuss about the different definitions and concept of sterilization and disinfection. Uh, Physical sterilizing agent we have to discuss, including dry heat, moist heat, different principle and working of different instrument which can be used in microbiological laboratory. Different chemical sterilizing agent, including phenolic substances, alcoholic substances, halogens. These are nothing but the different chemical substances which are used to sterilizing or disinfecting the microbiological uh, instrument or related substances. After that, we have to discuss about the pure culture techniques. What is the importance of pure culture? What is mean by pure culture, and how we have to isolate that pure culture by using different methods, including strip plate method, pour plate method, spread plate method. Again, we have to discuss about the basics of cultivation of microorganisms, including properties of good culture, different media used for the isolation of pure culture, role of buffers, definition and concepts of living media, non-living media, different types of media we have to discuss. Again, we will discuss different types of culture media, including different enrichment media, differentiating media we have to discuss. After that, we have to discuss about the different staining methods by which we can differentiate the microorganisms on the basis of coloring the body of that particular microorganisms. Different stains and dyes we have to use, different procedures and protocol we have to follow, so that we can isolate. After completing the syllabus, we can able to understand different disinfectant substances, sterilization method, so that we can kill or uh, maintain our particular environment uh, by avoiding the contamination of different microorganisms, so that we can culture different organism in the pure form. We can isolate that particular organisms from different environment in the pure form. We can use or able to understand different principle and workings of different instrument and equipment which are used in the microbiological laboratory. Different media preparation we can study, and by using that we can apply that particular protocol and methods to stain the different microorganisms, by which we can observe that particular microorganism under microscope. So in this we can discuss about the different stain, why we can use different stain in staining method, different dyes we can use, uh, different protocols are used for staining the microorganisms, including gram staining, endospore staining, flagella staining. So these are different methods, negative staining we can use in microbiological laboratory. So these are nothing but the basic aspect we can study in this paper. So let's start with your syllabus is nothing but the pure culture techniques. So firstly, one question remain in mind, that what is the need to isolate the pure culture, and what is meant by pure culture, and how that pure culture technique is developed. So first we will discuss about the historical background and what is the need and how the technique is developed. And now this technique is called as pure technique. So firstly we will discuss about the development of pure culture. Firstly we'll discuss about the background behind this. A population of organism cultivated in a medium is called as culture. So basically, while discussing about the microbiology, we have to know that microorganisms are ubiquitous in nature. They are present everywhere, but before or without growing them on a particular media, we could not observe or stain that particular organism. So that we need a bacteriological culture or bacteriological cell on a solid media to inoculate or to grow that particular organism. We need a different media, and when we culture that particular organism or inoculate that particular organism on nutrient media, we observe a different types of growth pattern of different organisms, and that growth is called as culture. So it is nothing but a population of organisms cultivated in a medium is called as culture. While a culture that contains only one species of organism is called as pure culture or a anexic culture, or a population of cells arising from a single cell is called as nothing but the pure culture. So basically, when we collected a different soil sample, we can say, or milk sample, or any another sample, and inoculated on a media, we can observe after 24 hour of incubation, we can observe different types of different colors and different size of colony on that particular media. So different color, different shape is nothing but the characteristics pattern of that particular organism, and they grow on that particular media. So depending upon the different colonical characteristics, we can differentiate that organism from one organism to another. So to inoculate particular or to observe or to obtain a particular single type of organism, we need a pure culture. So it is nothing but a growth of single type of organism which shows similar characteristics to one another. That organism or that culture is called as pure culture. So it is nothing but the, we can also say it is nothing but the anexic culture, or we can say it is nothing but a only one species of organism or one type of organism is growing that on that particular media is called as pure culture. So population of cells arising from a single cell is called as nothing but a pure culture. While another term we'll discuss in means of microbiological aspect, nothing but a mixed culture. Mixed culture is nothing but the rule of nature, meaning that the natural ecosystem like soil, sewage, milk, urine, it contains mixed population of several species of microorganisms. Several species, nothing but the different types of microorganisms are responsible or inhabit that particular ecosystem or that environment. So they are not visualized by the naked eyes, so that they are unable to grow only single type of organism without culturing that particular organism on a particular media. So that primarily we can observe a mixed type of culture. After that, we can able to inoculate or to isolate a single type of organism, and that culture is called as pure culture. So growth of multiple types of organisms on a particular media is called as mixed culture, or different species when grown in a particular media, we call that is nothing but a mixed culture. So let's uh, discuss about the what is mean by what is the historical background behind pure culture concept. So, firstly, the Antonio Han Leeuwenhoek, normally we called him as a nothing but a father of microbiology. He observed in 1863 first time a mixed flora in a natural samples like feces, urine sample, sewage sample. So, Antonio Han, Sir Antonio Han Leeuwenhoek, in 1863 he observed a mixed type of micro culture from sample taken from the different sewage samples, urine samples, or feces samples. You observe different types of organism, and he called there is nothing but a different types of microorganism. It is nothing but the mixed culture. In the earliest period, microbiologists had faced many problems because of contamination during their research. Later on, Sir Joseph Lister, who called as nothing but the pioneer of aseptic surgery, or in other words, we can say it is nothing but the father of aseptic surgery, first time developed a method of isolation of single desired bacteria in a pure form by successive dilation of a simple sample using a sterile fluid. So when Antonio van Leeuwenhoek observed different types of organisms, so he given a first proof that microorganisms are present in different samples. After that, researchers are unable to grow that particular organism or isolate. They observe multiple organisms at different generation time. They shows multiple colonies on particular media. So it could be quite be difficult to isolate and study that particular single type of organism. After that, Sir Joseph Lister observed or isolated a single type of organism by performing the serial dilution method, and he obtained a single type of organism, and he called is nothing but the pure type of culture of that particular organism. Later on, in 1872, a microbiologist, Scrocher, observed a growth in the form of compact masses of various colors on the slice of decaying potato. While when he observed such masses microscopically, he found that each mass contains single type of organism. Thus, he gave a clue that it is possible to isolate the organism in pure form by the use of solid media. When successive dilution are carried out by the Sir Joseph Lister, he able to isolate a pure single type of organism by carried out multiple serial dilution. But after that, in 1872, another scientist he observed a single type of growth of organism on a decaying potato. And when he observed that particular growth under microscope, he observed that the cellular morphology and characteristics of that particular organism are similar. That means they are belongs to the single species. So he knows that, or he get a clue that organisms are can be isolated in the pure form on the solid media. Later on, the different solid media or media preparation are developed. Then 1880, Robert Koch succeeded in using gelatin as a solidifying agent. Uh, Robert Koch is nothing but another scientist who have carried out a different research and most valuable work in microbiology in the field of bacteriology. So while working on the laboratory, he observed that gelatin can be used as a solidifying media to solidify the particular nutrient media. So nutrient solution with 5 to 10% gelatin can be used as a solid medium for the growth of organism. But it has a drawback as in summer days gelatin melt, so it could not be work as a solidifying agent. While some of the organisms which are able to liquefy or utilize gelatin as a source of nutrient, so that if gelatin is removed from that particular nutrient media, it could not remain in the solid form. So that though the cultures are isolated in the pure form, they become mixed culture. By that time, many scientists have gathered to Koch lab from all over the world. Dr. Walter Hess was one of them who was working with airborne organisms. He was frustrated by gelatin's nature. At that time, his wife, Fran Finney Hess, had suggested her husband to use agar-agar as a solidifying agent instead of gelatin. Agar is a polysaccharide derived from the seaweed. At that time, it was used for stabilizing jellies, thickening of soap. Agar was found to be as a perfect solidifying agent. Since then, up to date, agar is successfully used as a solidifying agent. Therefore, isolation of pure culture is nothing but the gain of isolated colony of desired organism on the surface of agar. Before that, one fact should be kept in mind that a single colony is formed from the population of single organism. So it is nothing but the historical background. To discuss that, Robert Koch carried out his work in laboratory and he found or he used gelatin as a solidifying agent for the solidifying particular nutrient media. But due to some drawbacks, he observed some difficulty to maintain the pure culture. So that different scientists gather and they discuss and they use agar-agar as a solidifying media, solidifying agent, which can be which overcome the drawback over gelatin as it could not be liquefied uh, before 45 degrees Celsius, and no one organism are still reported which are able to utilize agar-agar as a source of carbon. So, or sort of the nutrient media, so that uh, till date we can use agar-agar as a solidifying agent to solidify the particular nutrient media, and due to that we are able to isolate pure culture from the environment, though from the mixed culture. So it is nothing but the historical background behind the pure culture technique. Different methods are used for the isolation of pure culture. We will discuss in next lecture.